Learn about the newest digital pathology trends in science and industry, meet the most interesting people in the niche, and gain insights relevant to your own projects. Here is where pathology meets computer science. You are listening to the Digital Pathology Podcast with your host, Dr. Alexandra Zhurov.
Welcome to the Digital Pathology Podcast. Today, my guest is Victor Dillard. He's the Commercial Operation Director of Owkin. Owkin is a company that is active in different areas, but one of the areas is digital pathology, and that's what we're going to focus on. So welcome, Victor, to the podcast. Thank you so much for joining me. How are you?
It's a pleasure. Yeah, I'm doing very well, thanks. Great to be here.
Nice. Tell the listeners who you are and tell them a little bit more about Owkin.
Yeah, with pleasure. So my background actually is in chemical engineering, and I started my career in entrepreneurship, working for Flagship Ventures as a fellow in Boston, after which my own company in developing software solutions for CRISPR gene editing. And all of that was based on artificial intelligence. I grew that company and exited in 2019. And I learned a ton about how AI could be useful in fundamental and preclinical research. And all the work that we did was focused on basically R&D, everything leading up to IND filings. Mm-hmm. And so having spent the first part of my career basically in preclinical R&D, I really wanted to broaden my experience and get some experience into more the clinical development side. And I joined Owkin in January this year after meeting a lot of different AI teams in the space. And I was really impressed after being introduced to Owkin by the high caliber of people, the vision and the quality of the science, and the tech stack, which enables this awesome privacy preserving federated learning that I'm happy to talk about. And so I joined Owkin as commercial operations director. And what that means is I support our commercial and sales activities with processes and systems to structure that and all the reporting that goes around it. And a lot of my job is also to look at the technologies that come out of our lab and turn them and shape them into commercial offerings. So that means talking to early adopter customers, setting up pilot projects, and gathering that early feedback that helps inform the development before we then really formalize a pricing sheet and a method of deployment. And so a lot of my work is focused on doing that.
Okay, so what are the areas Owkin is active in?
It's not only pathology. So anything that kind of touches AI and clinical development, it falls under our mission umbrella. And so the way that we're structured is we have Owkin Lab, which is our R&D core, our R&D engine, and that's split into 4 different data modalities. So the first is histology. So we have a lot of digital pathology work that goes on within that team. And then we also work on radiology data, clinical data, and genomics. And in each of those teams, we have data scientists who are also paired up with medical experts or pathologists in the case of the pathology team, or radiologists, so actually, practicing radiologists who can really bring that expertise into the team. And then the kinds of projects that we try and work on are either projects that fall purely within one of those lab teams, or more excitingly, sometimes when they can actually leverage multiple lab teams. So multimodal or transversal projects where there's multiple types of clinical data.
Mm-hmm. And was it set up that way from the beginning, or did you move into pathology as a company at some point? How was that set up, and why there is also pathology? Because there is more to medicine and to clinical development than just those four categories that you meant. Why did pathology earn the place there?
I think pathology earned the place there because it's a really key part of a lot of modern medicine. And with the advent of digital pathology, it lends itself really nicely to AI, right? It's a perfect time to get into AI and pathology because we have now a much more and growing adoption of a digital pathology. So we have the images. And then there's AI technology that's been developed in image recognition, image to image translation. There's a ton of technology developments that allow us to actually look at pathology and say, okay, yeah, now we can do really amazing things to automate and simplify the pathologist's workflow, to accelerate or to even discover new biomarkers with the naked eye. it opens up a myriad of different opportunities. And so when our two co-founders, Thomas Clausel and Gilles Wainwright, came together and they were looking at where does AI fit within transforming medicine today, I think pathology was definitely top of the list because we have the images and we have the algorithms to work on them. Same thing with radiology. And of course, structured clinical data is always the starting point. And then genomics, also being now a digital science, if you will, lends itself very nicely to AI.
And how is the pathology team structured? And I tell you why I'm asking, because I want to ask how you work with pathologists. Do you have people on staff or do you have collaborations? What's the role of pathologists in this pathology offering?
Yep, both actually. So we have pathologists who are collaborators. So I'll tell you a little bit more maybe about how Owkin operates and then it'll contextualize it. So Owkin sets up partnerships with top tier academic institutes and key opinion leaders to build machine learning algorithms in collaboration with KOLs. And so these KOLs provide obviously the expertise, and they usually also then provide a data set that comes from their research institution. And then Owkin brings the machine learning, and together we build these fantastic models. And that's how the virtual staining that we'll talk about later came about. And then some of these KOLs have an extended relationship then with Owkin, and also to advise on specific projects outside of what we will have worked with them directly on. And so we will have expert pathologists in our network that are either part of our larger KOL network that we can go and talk to and get advice, or specific pathologists that are also acting on as a part-time consultant to Owkin. And then within the pathology team or the radiology team, we will also have a full-time expert. So in the radiology, we have a full-time radiologist. In the pathology, I think we have a part-time pathologist. That's the way that we operate, and really trying to bring the two minds together, because a data scientist needs an expert to work alongside them, and vice versa.
So you mentioned the virtual staining. This is the product or service. Is it a product or a service at the moment that you're offering?
That's part of my job, is that as part of the definition. It's an algorithm, and right now it's available on a license basis. So it's a product that you can license, and I can tell you a little bit more about it. It's a very kind of a curious new product. The idea is that we think about it as actually a technology platform, because what we started with are two markers that we think prove the concept that we can expand on this. But it takes an H&E whole slide image, and without having to do any real staining, it will create the digital immunostained whole slide image just from the back of the H&E slide. And we launched this with two IHC markers, a CD3 for staining lymphocytes and AE1, AE3 for staining epithelial cells. And so from the end user's perspective, the way that it works is pretty easy. It's the platform. We either have it on a server as an algorithm or within our software called Studio, which has a user interface. And the platform ingests an H&E slide in SVF or TIFF format, for example. And just using that, the algorithm will then generate a new image as a file on the server with the IHC marker selected. So that then medical researchers and pathologists can create thousands of digitized immunostained slides just from the H&E images at the click of a button. And on a normal computer, the whole process, I would say, on a single GPU takes on average 10 minutes to generate, a whole slide. So not just a tile or a section, but the whole image. And if you have a more advanced computing capacity and some of the fancy new servers, this can take a couple of seconds.
Okay, per slide.
Per slide, yeah.
So basically, it's instead of doing IHC and buying antibodies and validating this assay, you would take the algorithm and use it on whole slide images, H&E, and validate this process instead of doing the wet work with IHC.
Yeah, that's the idea. And at the moment, the output is just the image, which you could then use to load into a cell counting or cell detection model that you already have, or software. But so yeah, the output is a file that you can download and browse, and ideally, which is just the same as the image you would have scanned if you had done the staining with an antibody marker that you would have bought and done in the wet lab.
And how did you come up with this idea?
Yeah, as with many of the innovative models we created at Owkin, it started from a strong research collaboration that I mentioned earlier with a pathologist from our Owkin Loop, which is this network of partner hospitals that we have. And one day, effectively short on time and budget to carry out some physical CD3 staining. So we simply asked the question, what if we could generate the IHC image digitally, purely from the H&E slide? Is that even possible? And basically that's how the whole idea was kicked off. After that, there were many hurdles to overcome, but that was what incepted the idea.
Okay. And can you tell a little bit about the development process of this?
Yeah.
How did you transition from wet to virtual?
Yep. Actually, so the first hurdle is to get the data, right? With any machine learning project, the data is key. And so we had to generate 2 real images of the same slide, one stained with H&E, and then the other with, in this case, CD3, which was the marker we started for. So not consecutive cuts, but really the idea was to generate a whole slide stained with both markers, one after the other. And so effectively a double staining, right? So take the first slide, stain it, then wash it, and then restain it with another one, so that we can have, because if we did consecutive cuts, for example, we'd have 3 or 4 micrometers difference, which means that the exact cells in the whole slide image would change, and we would not be able to really train our model to because I'll get on that in a second, it's a model that does an image translation. So we needed to have the 2 perfect images. So we had to create this double staining protocol effectively to generate 2 images that we could overlay. The second hurdle is then the alignment of the 2 images so that they match perfectly cell to cell. So it's not a trivial task, because it's, I don't know, imagine aligning 2 satellite images of a city taken at different times and in different weather conditions. There's maybe some minor changes that are difficult to pick up and to align. And so we wanted to make sure that the whole image really was aligned overall, but then also at a patch level to have perfect alignment. So that was the second hurdle that we had to overcome. And that's where a lot of the data science work went into, was to really make sure that the co-registration was spot on. So then once we had those double stained aligned data, we used these machine learning generative adversarial network, or GAN, to train our virtual staining model. Specifically, we used one called Pix2Pix, which is typically used actually to, for example, add color to old black and white photos. And GANs are a pretty recent development, I would say, in machine learning. They're composed of 2 networks that compete against each other. So the first one is called the generator, and the second one is called the discriminator. So the generator, as the name suggests, creates these new CD3 images, and then it gives them to the discriminator, who then decides whether the image that was generated is real or fake by comparing it to the original real stained CD3, and then informs the generator whether it basically passed or failed the test. And then the generator iterates. And round they go until the discriminator can no longer recognize a fake image from the real one, because the generator has become so good at creating these images that it can effectively trick, if you will, the discriminator. So if you were to take the analogy of a painting, for example, if you wanted to copy a Picasso painting, you'd be the generator, and then me, the Picasso expert, would be the discriminator and tell you whether your painting is a real Picasso or not. And then after thousands of cycles, you get so good at replicating Picasso that I can't tell the difference. And so that was the use of GANs. I think another interesting aspect of what we had to do is that, these image generation algorithms like Pix2Pix usually work with images of about 1,000 pixels wide. So we had to adapt Pix2Pix by dividing the whole slide image into these tiles of 500 by 512 pixels. And that actually was really helpful, because it allowed us to have like about 25,000 tiles to train the algorithm from just one image. So from a data perspective, we get like really rich data from a whole slide image. And then so we could then also be a bit creative and add boosting features to train the algorithm more effectively by, let's say, spiking in some variety, like the images, or adding some color augmentation, so that the algorithm can be used and can get trained with a bit of noise and so it's not perfect slide by slide. And we cut the slides into these tiles, and then it randomly picks the tiles to learn from. But yeah, okay. So that's the process of creation.
Let me just sum up and correct me if I'm wrong in summing this up. So it's 2 networks working at the same time. And the one is generating images out of nowhere, so to say, and then improving the generation based on the feedback of the other network. of the discriminator.
Yep.
And then it gets more feedback and gets trained, whereas the discriminator has labeled data because it has the original IHC image. So it's like supervised feedback to an unsupervised image generation. Can it be?
Yeah, effectively. That would be one way of putting it. Yeah.
Okay. Great. So who is this for?
Again,
or who do you want it to be for? Because what stage of the project are you now?
Yeah, I think that's a useful point to start.
Yeah, so that I could buy off the shelf.
It is, but let me maybe give a bit more information around the performance of it, and then that helps people situate a little bit about what stage it's at. We assessed we trained today 3 different markers, as I said. So 2 of them, we are in advanced stages of that, which are the CD3 and the epithelial markers. And then we also have, which is just being finalized at the moment. And so when we finished training those models, we assessed the performance by comparing the virtually stained image back to the ground truth. And we look at the correlation between both images. In the case of CD3, we looked at the lymphocyte density, for example. When we take the correlation coefficient for the CD3 marker, we've got about 96% correlation between the 2 images. And in the case of the epithelial one, it was 97%. So we think that's pretty good for first generation technology. And the other test that we did is we gave the virtually stained image to our consultant, the pathologist, and they were not able to tell the 2 images apart. Tau by tau, a random kind of test, and they couldn't really differentiate. But for some, they could, but on the whole part, quite a difficult task. So we then also looked at at a cellular level, when we run, let's say, a positive cell detection algorithm on both images and then compare the results, we obtain precision and 72% and a recall rate of 63 for CD3. We think we can actually still improve on those results because we think those are important metrics as well, with additional data, as well as better cell detection algorithms, because they're not perfect either, as we know. And so we invite all these and continue to invite academic collaborators. So based on those metrics, we think that the algorithm is ready for folks who work in computational pathology or digital pathology teams within pharmaceutical and biotech companies and clinical teams, for example, who may want to run some proof of concept studies or want to explore retrospective cohorts of H&E slides for which they may not have the CD3 stain or the epithelial stain, to be able to maybe run some analysis and see whether or not there's a correlation of interest there. So we think that the technology is at a stage right now that for some of these markers, from an R&D perspective, they're ready to go, and you can come and license them and use them onto your images. But obviously, from a clinical perspective, we're still a long way away from validating the algorithm, and we're required to do a really rigorous study for that.
So if there's anyone listening that would be interested, they should contact you.
Yeah, absolutely. We'd be delighted to speak to them. Yeah.
Interested to take this to the next level or apply. And I will link the contact information in the show notes as well. So apart from this virtual staining, are you working on anything else in digital pathology? Or do you have your eyes set on something as the next project once this is totally mature? And
I wish life was I wish life was as easy for us as one project after the other. Owkin is now a 100-person company, and so there's got a lot of projects happening in parallel, lots of really exciting things. And in pathology especially, it's hard to be objective, I think, because we love all the pathology projects that we've got. So I'll pick out a few, but maybe I'll just focus on the track record that we have. I think one of the exciting work that we did was in mesothelioma, which was published in Nature Medicine just last year, where we showed by combining digital pathology with artificial intelligence, we could accurately predict prognosis for mesothelioma patients. But not just that, we also were able to discover new biomarkers that predict the response to treatments for mesothelioma. So we think that's a really great kind of case study for the capabilities of our pathology team, especially when we then bring in also the clinical data team and the 2 teams work together on these projects. The other side of what we're working on is we published also recently a model that's called HE2 RNA. And so what HE2 RNA does, it's another great invention from our pathology team. It basically predicts RNA-seq signatures from H&E slides. I think the whole sector of genomic prediction from images is really just starting to open up, I think, and Owkin is really pioneering in this field. We're excited about this. But so you can think about an RNA-seq profile like HRD, for example, that we could predict just starting from an HRD image. And then that can then be used to inform, let's say, patient identification or treatment decisions. So that's another area that we're innovating in and excited about some of the work that we're doing there.
I definitely want to link those publications as well in the show notes.
Mm-hmm. So. Yeah, and there's a whole page on our website with all the publications, but I think I'll send you the ones with the HE2RNA. It's very cool stuff, and we're excited about some of the more. It's a publication that talks about the HE2RNA model as a whole, and then we're excited to then come out with some of the really specific examples of how it works on particular signatures.
Mm-hmm. So you say for the virtual staining, you're using GANs, the generative adversarial networks, and they are pretty new. Before it was mostly convolutional neural networks, still is for the supervised learning. Where does this information come from in terms of innovation? Who comes up with this idea? Let's use this AI method. So then I'm going to ask you another question. Yeah. What about the pathology part of the innovation? But that's like more my part of the story. So that's why I'm starting with
I think I'll answer both at once, because at Owkin, I think one of the real fundamental beliefs is that innovation is collaborative, and really groundbreaking research is collaborative. And so this is why a lot of our work is done in partnership with key opinion leaders who bring that expertise from the hospital, from the pathology lab, from the radiology lab. And then Owkin brings the machine learning expertise within individual disciplines, as well as a really good and deep understanding of the clinical landscape around that, so that we're able to have a common language to speak with the KOLs and the experts. But it always usually starts with the partnership, a very close partnership with our academic partners, as well as the lab that I said, our R&D engine. It's a world-class data science team. One of our co-founders was a professor in machine learning. These are the experts, and they're at the forefront of machine learning techniques, and this is what they do. And they go to machine learning conferences and they read machine learning papers all the time. So this is their expertise, it's their job. They're very good at it. I think they have that information, but your question is really around how does that innovation happen? For me, it happens when the collaborations come together and the experts challenge each other and also ask questions that trigger things that then make the connections in your brain that allow you to say, actually, yeah, we should try this. It doesn't happen in silo.
I think it's a very important point that you have to have an understanding of both disciplines on both sides. You're just expert in one, but if you don't have enough understanding of the other side, you don't know which methods you can apply. So if you don't have enough understanding of how IHC works in the lab, you will not be able to apply the appropriate machine learning method to generate it digitally or virtually. So that's my mission as well of the blog and of the podcast, to bring those two worlds together.
Yeah. So, I think one of the things also sometimes with machine learning for people who aren't in the space is that it's a pretty black box approach, right? Where people just say, hey, okay, I've got this algorithm, it takes this stuff, and then it outputs this magical result, and we don't really go into detail about how it does that. I think a key part of, again, it's core to our mission here at Owkin, is to make machine learning understandable, and we believe in a clear box approach, where actually when we get a result, we can go back and understand how that result was generated. It doesn't apply itself necessarily or lend itself easily to every type of machine learning, but if you're going to develop an algorithm that's going to be used by scientists or ultimately by clinicians, you need to be able to explain it, and you need to be able to open it up and build that trust and that understanding that there's a logic to how the result is generated. And oftentimes that actually is a really interesting part of the whole conversation, is how did it get to that result? That's often more important than the result itself.
Definitely. I think that also increases the credibility of the method if you can see what's actually happening. And now that it's also required by the regulators, at least in European Union, the data protection regulation requires this. So if ever this is going to be applied to decisions on humans, it has to be explainable. And I have talked about it, I think, in some other episode, that there is this whole new emerging area of explainable AI. That's exactly what's growing on top of the machine learning movement, if you can say that, machine learning scientific discipline. There's this another sub-discipline that tries to explain. So it's great that you're embracing this. You'll have to, because you're located in Europe, right? Okay, where you are and where you were.
Yeah, well, Owkin it's now a global company, but our R&D center is in Paris. So that's really where the majority of our team is located. And then in the beautiful city of Nantes as well, in the west of France. But our corporate headquarters are in New York. And me personally, I work from the London office. And we just finished our Series A, which brings a total Series A round to 70 million. So it's really a great time to join Owkin. We're in full expansion. We'll be in Switzerland soon. I think it's hard to, when people ask me, where are you guys located now? It's the trend now. We can work remotely, that's the day and age that we live in now. So that's not a problem. But also Owkin is really focused on hiring the right talent for the job. And if the talent is in San Francisco, or if it's in Berlin, or if it's in Madrid, then, we will adapt to people's situations. And I think that's a key part of the mission of growing the team.
So your R&D lab, this is not a wet lab. This is a machine learning and computer lab.
Yeah, exactly. That's based in Paris. And then any wet lab activity is usually done by the academic collaborators that we work with.
By the partners. Okay, So what was the most difficult part in this virtual staining development? What was the thing? Was there something that you did not expect, like the major hurdle that you had? You mentioned some of them, like the aligning of slides.
I think it depends on who you speak to within the company, because of course the data scientists working directly on this, they might say that part of the alignment was particularly tricky, but I think as a whole process overall, one lesson that you always take away is you want to engage not just the expert pathologists early on, but you also want to engage your customers as quickly and as early on in the process. And they will give you ideas about, let's say, I don't know, which markers are more interesting to them. We chose CD3 because we had the data there, and as an AI company, it's always a balance of, if I've got the data, I can start and I could do the proof of concept, or do I try and find the perfect data and then delay? So here we chose CD3 to validate it. But I think as a learning, it's always around how quickly can you engage your customers and get that feedback about which markers are interesting for them. Do they prefer the fluorescent markers, for example? So should we adapt our machine learning to maybe work with fluoro and multiplex systems, and that kind of information around where should we take this once we've got the proof of concept, I think is always a key learning on how quickly you can generate that feedback loop.
I guess it's also crucial now that you don't get stuck in your idea, because if everybody uses it, then you had a nice idea and that's it. You cannot really build a real life business around it.
But it's not always super easy, because sometimes the people don't necessarily see the application right away. And sometimes maybe with virtual staining, once we get 5 or 10 markers available, then suddenly you've got a whole multiplexing capability that you can do straight onto your computer that you didn't need to do before. And maybe some people will come in and say, oh, actually, this triggers a really great idea for me. I think we could go ahead and look at this whole panel of things that would have taken me a ton of time to do manually. Again, it's part of that iterative cycle of innovating. You've got to keep in touch with the market and your customers, but also continue pushing on the vision and taking risks. And right now we continue to take the risk that we believe this is a great platform and it needs to continue progressing. And even if not every marker works, we learn a lot about machine learning at the same time. We learn a lot about digital pathology workflows and particular markers. So it's certainly not a lot of knowledge that's wasted or lost.
I think there's definitely a lot of application for this. I think that one hurdle will come from the skepticism of the future applicants of this. And here this explainable AI comes into play. I think because IHC as such is a complex method, there is a lot of variables that influence the binding of the antibodies. And I think there's going to be significant challenge to convince people that it can actually be done digitally. That's such a complex procedure. Interaction between the molecules in the tissue can just be translated digitally from the information in the image. But if you have this validated sufficiently for different markers, and you can point out to what's being the driving factor in deciding that these cells are positive. And it doesn't have to be something that we see with our eyes, because if it was, then probably we would have already seen it.
Yeah, that's the debate internally. I think there will certainly be markers for which the model and the platform will not necessarily perform with sufficient accuracy that it can be deployed.
It's fine. It doesn't have to be everything.
It does exactly. And so we hope that at least for the ones that visually we can distinguish from an H&E slide, for that we can definitely hammer it out of the park and have a great performance algorithm. And then the other side of it, which we think is exciting, is maybe there are morphologies that we're not necessarily aware of right now, or distinctions that we're not aware of right now to the naked eye are too small to distinguish, but that the algorithm will be able to detect, and that maybe it doesn't yield a super high performance, but that's linking it back to the explainable AIs. If we can go back and understand how did it try to differentiate and do this marking, maybe we'll pick up some new things. So it's weird. I think that's part of why we want to test a range of markers, from what people might think are easy ones all the way to what are certainly going to be some of the more challenging ones, like probably PD1 will be an interesting one to go after.
Yeah. I am excited. I keep my fingers crossed for this to succeed and to get to the next level. Is there anything else you want to tell the listeners?
First of all, thanks for listening to me talk about Owkin and virtual staining. And if you have any interest in technology, or of course in learning how it was developed, Alex, I'll share with you the link so you can share it with your listeners. There's a blog post where our data scientist Olivier, who did the great work here, talks about this journey in developing the model. And then, of course, if you want to trial it, get in touch with us. We'd be more than happy to show you how it works, and also try it on some slides, H&E slides of your own. So we're open.
So one last question. You said it can be either sent as an algorithm to use on your own platform or within your software platform.
Yeah, as a kind of platform on which you could run this, there's really two ways. The first is you can imagine if you have a server in your institution, we can deploy the algorithm, and you can basically send an image via an API, an application programming interface, and then the algorithm will just run directly on your server and then generate the image, and then you just go and find the file directly there. So no user interface. It's much more of a command line approach. Owkin also is developing a software platform called Studio in parallel. And Studio is software that we develop for medical researchers who want to train new machine learning models. And so it works with pathology images. And just recently, we also added radiology as a functionality. And so you can build your cohort within Studio. So assemble your pathology images, for example, and then train a new machine learning model, or apply one of our existing models, like a prognostic model or a recognition of certain gene expression. And you could also do some virtual staining. So on Studio, soon to be released, get onto the platform and then click on the virtual staining, upload your H&E slides, and then you get the virtually stained slide to be able to download or just visualize and browse directly in Studio. So that's much more of a user interface. You can click around, and it's if you're not a software person who is comfortable with a command line, that would be the solution for you.
Okay. Thank you so much for joining me today. Thank you for taking your time. And have a great day.
Thanks, Alex. Have a great day, you too. Yes, all the best. Bye-bye.
Thank you for listening. For more great digital pathology resources, visit the Digital Pathology Consulting website and subscribe to our newsletter on digitalpathologyconsulting.com. After subscribing, you will get access to the free digital pathology crash course, which will help you start working on your digital pathology projects immediately. Talk to you in the next episode.